Testing. Aha, we're on. Excellent. Okay. Well, welcome everybody. Thank you for coming along. So, we okay? We got a bit of feedback. We've got some help this time. For those of you that have been here previously, sound has been challenging, but I'll wait for the thumbs up. Okay? Great. Okay, so yes, welcome to everyone today. Thank you for coming. Today is the third program update of an exciting BHP and Curtin University initiative centring on the field of environmental DNA, or eDNA, and its use as a biomonitoring tool. I'm Shane Herbert, and I'm the program manager for the initiative that the team will be presenting on today. So before we get started, I'd just like to make an acknowledgement. So Curtin University acknowledges all First Nations of this place we call Australia and recognise the many nations who have looked after country for more than 60,000 years. We're honoured and grateful for the privilege to maintain campuses operating in Boorloo in Perth and Karakula in Kalgoorlie in Australia. We pay our respects to elders past, present and custodians and owners of these lands. We recognise their deep knowledge and their cultural, spiritual and educational practices and aspire to learn and teach in partnership with them. Curtin also acknowledges First Nations people connected to our global campuses and our global collaborations. We're committed to working in partnership with all custodians and owners to strengthen and embed First Nations voices and perspectives in our decision making now and into the future. So I'd also like to address a few housekeeping matters. So in the event of emergency, please exit through the doors that you entered, so the stairs that you've just come up. There's also an elevator there that will bring you down to the ground floor. Don't go 2 stories down or you end up in a basement. But down to the ground floor, obviously if there's, in the case of fire, please don't use the elevator. Now the muster point is south through Brookfield Place, across the footbridge, over Mounts Bay Road, and you can see on the map there. We've got a bit of a walk, so hope you'll wear comfortable shoes. But that's the open space where we'll be gathering. Toilets are located through the door in the hallway off to my left there. Can also ask that you all take a moment to check your mobile phones. Good news, we've organised refreshments at the conclusion of these presentations at the print hall, which is not too far away. In fact, I've marked it on the map there. But we'll have a series of team members showing you how to get there. But in the meantime, we've got some water up the back, just on the black cloth up the back there, for anyone that wants a drink in the meantime. Yep. So this presentation is also being broadcast by WebEx. So all of that is less relevant to our online speakers, but we've muted the lines. So if you have any questions for the speakers, if you could pop them in the chat, and Cheryl Lee, my colleague up the back there, will put them to the speaker if we have time. We'll also be aiming to capture today's proceedings in a recording and make it available afterwards. So if anyone has any privacy concerns, if they could come and let me know, or someone from the Curtin team. All right, thank you for your understanding. I think with all that said, I'll move on to the talks. So here's today's agenda. Once I'm finished, I'll introduce Dr. Tim Cooper. And then we've got Professor Prof Allentoft, who unfortunately can't join us today, so he's provided a recording to introduce the program. Then the order of proceedings will be our Pilbara Olive Python Detection and Conservation project by Marsa and Ben, followed by Subterranean Fauna Detection with Dr. Nicole White and Michelle Guzik, etc. And then we've got the invasive species studies, which is actually 2 of the projects being presented by Professor Simon Jarman. And then we're introducing a new project by Dr. Mika van der Heijden about terrestrial ecosystem biomonitoring with eDNA. And then to finish off, we've got unveiling the functional ecology of hypersaline lakes through multidisciplinary approaches with Dr. Matthias Sacco and a Chilean student, Gonzalo Salazar. And then we'll have final remarks and we'll move on to drinks. So with that, I'll move on to introducing Dr. Tim Cooper to the stand, who's the Principal of Biodiversity and Land Management at BHP, to say a few words. Tim has been instrumental in this initiative and continues to be a strong guidance to the Curtin Uni team and to eDNA through the various channels that he's involved with. with his help, we've been able to navigate this program through the challenges of COVID and build a strong eDNA program that we continue to be excited about. So over to you, Tim. Thank you.
Thanks very much, Shane. I was just having a bit of a joke with Nicole. I'm only referred to as a doctor once a year, and that's at this meeting. So thank you very much for that honour. But yes, no, it's great to be back here again and to welcome you all to the Year 3 update of the global, the Environmental DNA Study for Global Environmental Studies, or the eDGES project, as we call it. I'd also like to begin by acknowledging the Whadjuk people of the Noongar Nation, the traditional custodians of the land and waters on which we're meeting today, and pay my respects to their elders past, present and emerging. So yeah, for those that don't know me, that's my title. I work in the biodiversity and land corporate team at BHP, and just move on, yes. So what do? We help drive the strategic direction for the company on topics such as biodiversity. And I've taken this image here on the left from our website to provide a picture of how projects like the eDGES project are relevant to the biodiversity work we do at BHP. We've got a vision for contributing to the global goals for nature, and partnerships like this one with Curtin are really important in helping us understand how we can use innovative techniques such as environmental DNA to measure changes in nature over time. And we know that there's the biodiversity that you can see, like in that landscape over there on the top right, but we're also really interested in the stuff that you can't see, as well as the cryptic species, to help us understand and provide a complete picture of biodiversity as we start thinking about how we can contribute to those global goals. So as Shane mentioned, the eDGES project is funded by our social investment project, and that's our voluntary contribution to projects which have the primary purpose of contributing to the resilience of the environment and the communities of which we are part. And it's delivered through local, regional, national and global projects. And this project is well and truly in that box. The eDGES project is by far the most productive social investment environmental project that we have. And I looked on the website this morning, we're up to 8 papers. And I'm sure there's numerous other papers that are in preparation. The team are also very active at national and international conferences. So we're getting some great reach through this project and through the team. So my thanks to you already. And I've also included a few photos here of another social investment project that we published recently on the application of natural capital accounting at a closed mine site. So that the landscape there in the top right is from a closed site in the southwest of Western Australia. And whilst we didn't use eDNA techniques in understanding the values on that site, we see great potential for novel ideas such as the tiki. And I'm hoping Sean Wilkinson has dialed in, or maybe he'll catch the recording. We see great potential for approaches like that to helping us understand how we can quantify spatial and temporal changes in nature through time. And we've got plenty of land where we can run pilots and test ideas. So look, thank you for taking the time out of your busy diaries to attend this afternoon. This is, like I said, this is one of our truly global projects, and I'm super excited by eDNA, the possibilities where it can go and how corporates like us can use it in our decision-making processes. So thank you, Shane. Back to you.
Yes.
Okay. Thank you. Thanks.
Cheers, Tim. Okay, so now we'll switch across to Morten. Do I just break out of this and, oh, there we go. Okay, so yes, Morten's got a presentation. And
good afternoon, everybody, and thank you so much for joining us here this afternoon, where we will be showing you some of the results and progress on our eDGES program. My name is Prof Allentoft, and I apologize that I couldn't be there in person this year, but I do know that you will be in very safe hands with Shane and the rest of the team for this afternoon. Here I will just be giving you a little introduction to the eDGES program, and then my colleagues will take over and show you more details and results from each individual project that is involved in this program. So the research we'll be presenting here today is taking place in Trent Lab at the Curtin University, the Trace and Environmental DNA Labs. Most of you will probably be familiar with these labs already. Here we have some wonderful resources available for carrying out our research, and we have a really good crowd of people for operating these labs to ensure that we are generating high quality research. One thing that is a little bit unusual about our labs is perhaps the access here to highly sterile clean lab environments. This is a big benefit when you're operating with the problematic degraded DNA that is prone to contamination. Here's a big benefit that you can keep things highly sterile. In our labs, we have a three-pronged approach to our research. We use environmental DNA to ask what biodiversity is present. We use ancient DNA to ask what biodiversity used to be present. And then we use genomics to ask what happened in the genomes, the evolutionary processes along the way. And by merging these three molecular disciplines, we can ask questions around the insights on the formation, distribution, and resilience of Australian biodiversity. Most of the research we'll be presenting here today and within the eDGES program will be centered on this component here, environmental DNA. That's not everything we do in Trend Lab, and it's not also everything we do within the eDGES. You will see elements of this genomics aspect here as well today. And we may in the future also merge in some ancient DNA research in the eDGES framework. Ancient DNA is very useful in this context because it can provide a baseline for understanding current ecosystem health. If you're just looking at a snapshot of the present, it may be difficult to actually evaluate the health of your current system. And here ancient DNA can provide a reference point for that. This is a nice little summary slide that simply kind of explains in broad terms what our goals are with eDGES. And I'm just going to read this up. We want to develop and apply environmental DNA technology to address biodiversity loss and sustainability. We seek to understand ecological and evolutionary changes in natural and man-made landscapes. We want to develop new generation tools for managing species. And managing in this context here means in a conservation-related context. We want to conduct biodiversity conservation through whole community studies. And as you've already heard, this program is obviously supported by BHP's social investment framework, which is the whole reason why we're here today, why this is even doable. With the research, when you have a research program like this, it's of course very important to establish some criteria for success. We have 6 projects in eDGES. Each of them builds and expands on environmental DNA's utility as a biomonitoring tool, and have an overriding aim of translating our academic knowledge into new outcomes. And those outcomes can be broadly divided into 4 different groupings. So first and foremost, this is a research project. We are all scientists, so it is very important and dear to us to produce high impact science and produce peer-reviewed papers. Of course, it shouldn't stop there. It's important. Environmental DNA in general is a very applied science. So in many environmental DNA projects, and in particular in the eDGES framework, it's important that this research knowledge is translated into direct conservation outcomes, such as new biomonitoring tools that we can actually apply in a conservation context. We, as part of any research program, at least where there's an international scope, you will of course facilitate networks, and for expanded larger studies, this is the collaborative aspects of having a big research program like this. And that's very important both for us as researchers, but also the students, PhD students, honor students, so forth, that we're actually training in a program like this. And that it doesn't only just benefit ourselves, but it benefits both future researchers and of course a broader international group of people. Then we have a community education and awareness aspect to this program, and this is what we're doing right now here today, for example. We are also looking at implementing a citizen science component moving forward into the eDGES framework, and that could be a nice way of implementing or getting the public more involved in the research we're doing. And then something we haven't dabbled too much with so far, but something that is clearly very important when you want to do applied research is input to policy. It shouldn't just stop with the scientific papers or community awareness. Somehow, some of these tools or some of the insights that we're creating should ideally be implemented into a political setting in order to really make a difference. So in summary, these are the four primary aspects where we hope eDGES will make a difference. And some of these aspects we're already making a difference. We are already publishing papers. We are already community educating and so forth. But these are the four main aspects where we hope to really make a difference with this program. Our eDGES program covers projects. There's 6 projects in total across 4 different environments. We have 2 projects that are focusing on biodiversity monitoring in terrestrial settings. We have 1 freshwater project that is looking at these saline lakes in South America. We have a project on subterranean fauna, and then we have 2 projects in a marine setting. So it's really a kind of a wide scope, at least when it comes to biomonitoring in different types of biomes. And should there be anyone in the audience by now who's not aware of environmental DNA, then this is a nice summary slide that kind of shows all the different applicabilities. So eDNA is, as the word says, DNA that exists in the environment, and it derives directly from the organisms that are existing in the environment. So all organisms will shed DNA into the environment, whether by defecation, urination, when they die and decompose, by shedding skin cells into the environment, and so forth. So constantly, organisms living in an ecosystem will deposit DNA into that ecosystem. And what does this mean? This means that when we could go out, we can take a water sample or soil sample, even an air sample. We can extract the DNA from those samples, and we can sequence the DNA, and then we can identify which species are living in that ecosystem. That's environmental DNA in a nutshell. So it obviously becomes a very important tool for biomonitoring. You can see here that there are many different applications. You can use it in sediment, in air, even as I mentioned, and in water, and of course in many different applied setting where you need to have information on the species that are living in the given area. So one thing that is very important to the eDGES program, and I should say science and trend lab in general, is that we are innovative, that we try to constantly push the technology to the limit and push into new research avenues that we haven't really considered before. And the eDGES is certainly one program where we're doing this. You will see that there are elements in all the individual projects that are highly innovative and that are testing new things, whether this is new sampling ways, new way to try and extract the DNA, new ways to try and sequence the DNA, new ways to try and analyze it. So there are innovative elements all over the eDGES framework. One thing that we are very excited about and that will really allow us to explore new methods in eDNA research is this new sequencer that we purchased just recently, heavily supported by BHP, I should say. This is the NextSeq 2000 system, and this will really allow us to generate a lot more data, and it will allow us to be testing new frameworks and new concepts in eDNA research. For example, you will hear shotgun sequencing mentioned several times today, and this is a way of not sticking to just the short 200, 300 base pair barcodes that we've been analyzing so far, but actually try to sequence the total amount of DNA that is in the environmental samples. And this has some exciting prospects, also some exciting challenges, I should say, but this is certainly something we will be exploring for years to come. So we're very excited about that. My last slide here is a fairly simple one. You can all see that it says just so what. And that is, of course, a very important question, in particular in the eDGES framework that has quite an applied angle to it. We are producing basic research. We are testing and inventing new methods, but of course, we need to ask ourselves the so what question all the time. It's important that we make a difference. And it's important that the research we're producing actually have an impact in relation to conservation management, for example, biomonitoring and conservation management. So you will hopefully see that throughout these presentations you're seeing today, there is a strong so what element to them. The previous years we've held this little seminar series here. We have kind of taken a little bit of an academic angle to it, presenting the results in a very academic way. But this year, I hope we've tried, and you will hopefully see that we've managed to do it a little bit differently, asking the so what question, trying to express in clearer terms why we think this is actually making an impact and what our aims are in terms of producing science that has an impact beyond just the research papers, of course. So, and please feel free to raise your hand and ask the so what question if you don't feel that this is properly explained when my colleagues are going through these individual research projects in a second. So I think that's all for me. Again, hope to see you all next year, and I'm sorry I couldn't be there in person today, but please enjoy the rest of your afternoon, and I hope you'll have a great social time as well when the talks are over. Thank you so much.
Great, thank you, Morten. Believe he's online. I think he may actually even be on a plane on his way over, actually, so back home. Okay, so now into the scientific presentation. So I'd like to welcome Dr. Mousavi, there she is, who will also be talking with Mr. Ben Heyward on Pilbara Olive Pythons.
Hello and good afternoon, everyone. Thank you for being here today. And I would like to talk about the first eDGES program, which is about eDNA biomonitoring and also conservation genomics of the Pilbara Olive Python. So we know that olive pythons, they are one of the world's largest snakes. They can grow up to 6.5 meters. They are very expert swimmers because they actually use the water holes as a place to hide and ambush for their prey. We do have two subspecies of olive pythons in Australia. So we have the Kimberley subpopulation, which has a wider distribution, and we have the Pilbara olive pythons, which are pretty much restricted to the Pilbara region. And they are vulnerable to extinction, and they are also difficult to biomonitor with traditional methods, which is why in the eDGES project #1, our aim is to develop high-precision eDNA tools that can help us in basically monitoring the species in the field. And also, we would like to develop a high-quality reference genome for the subspecies, which can be used as a foundation for large-scale evolutionary studies, which we are going to do down the track. So we know that they are using the rock pool waters for hunting. And so our first step was to figure out, can we actually, using eDNA metabarcoding methods, actually are we able to find them in the rock pool's water? And why is that even a question? Because reptiles are a difficult target for eDNA type of studies. And the reason behind that is that they have this keratinized skin and also a slower metabolic rate, which means they're going to shed less DNA into the environment. Hence, it's going to be harder to pick them using our eDNA metabarcoding assays. We also, as a first step, wanted to develop a custom database, a reference database for 8 species of pythons that occur in Australia, because one of our steps doing eDNA metabarcoding is to eventually compare what we found with a custom database, and based on that, we can do assignments. So you can guess that if what we are looking for does not exist in the custom database, then we are not going to find it. And some of you may remember that last year we have presented some preliminary results in this regard, and we are very proud to announce that that preliminary results has actually been published this year, and you can go and have a more detailed look at it. But I'm just going to show you some quickly, just to recap on some of the aspects of that project. Thanks to a few consultation companies and their clients, we were able to collate a total of 228 water samples from across the Pilbara region, which was a part of an ongoing biomonitoring in that region from 6 different biomonitoring zones. And so those samples were collected from 40 rock pools, and then through our normal eDNA metabarcoding steps, we used a reptile assay to basically see if we can detect the olive python, Pilbara olive python or not. Also, as part of this project, as we promised, we developed a custom reference database, which included 39 sequences from those 8 different python species. And that database included sequence samples from both Pilbara olive python and the Kimberley olive python. So what did we find? Plenty of everything, including 2 sequences that were assigned to 2 snakes, but unfortunately, not the snakes that we expected to. So as you can see, one of those species does not even occur in Australia. It is actually native to Southeast Asia. But searching against the general public database at percentage identity of 98 and 99, these were the 2 snakes that we could actually match the sequences. And this is where the importance of the custom database I talked about comes to play a role, because thanks to that custom database, then we were able to reassign confidently at 100% identity back to the Pilbara olive python, which is what we were looking for. Sorry, this is not moving. Okay, fix it. Sorry. Okay. So the second aim that I mentioned was to develop a high quality reference genome. And so a couple of years ago, with the help of PERSU experts, we sampled blood from this female olive python at PERSU, which then we sent it to the Vertebrate Genome Lab in the US, where they have this really high-end, state-of-the-art technologies that can enable us to have a complete reference genome. But what is a reference genome? How is it built? And what steps are involved in that process? So it starts with getting a high-quality sample, in this case, blood sample, which we then extract the DNA from. And then that DNA goes through fragmentation, so it becomes a lot of smaller pieces. Then those smaller pieces, they get sequenced. And once they are sequenced using computer programs, we can look for the regions that are overlapping. And then using that information, we can start building longer sequences, which we call contigs. So they are like a continuous sequences. And then if we do have access to those high-end technologies, which in this case we did, then we can even go beyond that and make that continuous sequence even longer and put it into what we call a scaffold. And eventually what we really want is that that scaffold becomes so long that it can cover the whole length of a chromosome. And that way we can say, okay, this is pretty much of a good reference genome. And so what happened? We do now have this beautiful, complete reference genome, which is 1.5 gigabase per, and it was put into 17 chromosomes. So, and some of these numbers that you see, it essentially, what it's trying to say is that we do have a great assembly and it is of high quality and the contiguity of the assembly. But why is that even important? Why do we need to have a reference genome? So a reference genome is a base for everything that we want to do downstream, because that allows us to then go into the field and sample individual snakes that we can then resequence, map it back to the reference genome that we have, And through that, we can compare these different populations for very important aspects, such as what is the level of genetic diversity in these populations? What is the inbreeding level? We can even figure out how these different populations are being affected by climate changes, for example. Also, using this reference genome as a base, now we can actually step beyond eDNA in its traditional way, because now we can use this reference genome to then go and look for genome-wide trace of Python DNA in our environmental sample, which I will explain a little bit more shortly. But what all of these things mean, having these scientific facts in place, that is what is going to help us to eventually be able to refine our conservation and management strategies. So we now have this beautiful, complete olive python reference genome. And so what? So what currently we are using in the field of eDNA metabarcoding is only using a very small proportion of that information. But you can imagine in an environmental sample, it is not that little short barcode, which generally speaking, it is going to be below 300 base pair in length. In an environmental sample, it is not only that 300 base pair which is floating, but it's the whole genome information in there. And simply, it means currently we are only using a very, very small portion of the genomic information available in a sample. And this is where the novelty of our research comes in, because of what we are planning to do is now trying to use other type of methods that can enable us to now go beyond that very small portion, and we can recover more genome-wide information. And that is going to be done initially through shotgun sequencing. And what does that mean? So again, we do have a DNA, which is this time it's coming from our environmental sample. So it's not only the Python DNA in there, but all the other organisms as well. And then this DNA, extracted DNA, will get fragmented and sequenced again. And now this time, because we do have the olive python reference genome, we can look for areas, we can map those reads and see, can we actually find and match them against our reference genome? And this is what allows us to basically collect that genome-wide information and go through using that information, which is something that our PhD student Ben is going to discuss a little bit more. But what did we do? So we did run a pilot study in this regard, and that is built based on what Ben has done during his honors study last year. And to recap on that study, you may remember, so he had a tank experiment. There was 2 tanks, one water from lake and one ultra pure water, which then python blood was spiked into the 2 tanks, and then at different time points, samples were taken. And what he wanted to study back then was to figure out what is the decay rate in these scenarios. And just to quickly remind you of that result, so he could show that the DNA decay rate was much faster in the lake sample. And in fact, in a matter of a couple of days, most of the Python DNA was gone in the lake sample. So what did we do? We actually picked a few time points from that study, which we shotgun sequenced and mapped it against the reference genome that we now have. And this is basically what we got. So just to quickly talk about some aspects of it, as we would expect in the ultra pure water, we got a lot higher number of reads being mapped to the olive python genome, versus in the lake water, we do have a lot less. And one thing that is again in line with what Ben already discovered is that you can see at a time of 0, which is exactly when he spiked the tanks with blood, he's getting the most reads, and then it quickly decline. So obviously this is only a pilot study and it's just the base for what Ben is going to discover and look into more deeply during his PhD studies. And I would like to invite him to join me and talk about his PhD studies now.
Thank you, Masa. So in my PhD, I will have 4 main data chapters to achieve these aims over the remainder of the project. And these will be data chapter 1 as the development of a precision standardized species-specific probe-based assay. This will allow us to validate the presence of Pilbara olive pythons in any samples and in the future methods that we use. Data chapter 2 is this genomic eDNA decay. So this will have me enhancing the results I got from my honors, but this time we're not going to look at that small region, but over the whole genome. And this is really cool because it will tell us which regions are resilient and which regions we can find over time. So it's very special. Data chapter 3 is this eGenomics framework. So this is me developing and validating this whole genome biomonitoring. This is a really novel use of the whole genome, a really novel aspect of the project. So I'll tell you a bit about it in more detail. And finally, data chapter 4 is the population genomics. So in there, I will be going out into the field, taking individual Python samples, and then resequencing them to obtain high quality genomic data. This is critical for conservation because if we don't know where they are or how they're faring, we can't conserve them. So that's a really focal aspect as well. Some particularly novel aspects of this project include data chapter 2, this genomic eDNA decay. Because while we know that genome regions decay at different rates, and it's suspected that this has to do with the actual characteristics, how the DNA is packaged or the structure of the DNA, it's very poorly studied, particularly in an eDNA context. So how the genome, the behavior of the genome in the environment over time. Understanding this would not only help us elucidate the behavior of the genome, but also highlight which regions are resilient, which regions we can target, and that will be in the environment for longer. So that's a really cool bit. And chapter 3, this eGenomics chapter. This is a really focal chapter because as Marcia mentioned earlier, it's possible to get the whole genome's worth of DNA from the environment, but this has yet to be done for a targeted terrestrial organism. So, and what has been done is marine and in the past year or so. So it's a really emerging, very cool part of it. So we will take, we need to validate the method and we will do so at Perth Zoo and Caversham Wildlife Park, where they have olive python enclosures. And we will sample water, wooden rock surfaces, soil, and air. And then we will take these through shotgun sequencing with our in-house NextSeq 2000, and then mapping to our Pilbara olive python reference genome. This validation will allow us to determine the most effective substrate for Pilbara olive python DNA and which functions the best. And then we can take that into the field and use it on actual rock pools. Over the course of this project, we will look to enhance Pilbara olive python biomonitoring and further our understanding of the Pilbara olive python, not only through improved biomonitoring tools, such as the qPCR assay, but also through pushing from eDNA techniques to eGenomics, so the whole genome. And with this, we can consider whole genome information directly from the environment. With the Pilbara olive python, a model organism, this will enable population level studies and future-proof such eDNA techniques as sequencing becomes cheaper and bioinformatics capabilities improve, it'll improve how it's used, and more people will be using it. And of course, population genomics that will allow us to assess the population structure, the genetic diversity and the inbreeding, and any selection pressures that these pythons face in the Pilbara. With all this together, we can thus refine conservation management techniques and the management plans of this vulnerable python. Thank you.
Is this on? It is on. Okay. Thank you very much, both speakers there. Does anyone have a burning question? We're right on time.
Great work, guys. Have there been any attempt to use fecal matter as another possibility, or not only to identify the species, but also for dietary studies?
Not yet. We have not tried that, but it's something that, not necessarily for this project maybe, but it's something that we do have the capabilities in the lab, and we did try it with other species in the lab, but not in particular for python yet.
Yeah. Okay. Well, we might move on if that's okay. We're right on time. So our next speaker is Dr. Nicole White and Dr. Michelle Guzik from the University of Adelaide, who are tag teaming today, talking about subterranean fauna conservation and detections. Over to you.
Yeah, you can come up. Good afternoon, everybody. It's great to see so many familiar faces and some new faces, so welcome. You probably heard about eDGES. I'm excited to share with you the Subterranean project that we have going with eDGES. So originally, when this project was proposed, it was a fairly small project with the aim, we've been hearing about databases, the importance of databases. I had this aim of kind of moving beyond the barcode. I'll explain what the barcode is. And then we had COVID, and that changed a few things. And we actually produced a bigger project from this, which is what I'm happy to share with you today. But before I go into my bit, and this is how Michelle comes in, I'd like to officially introduce Michelle. She's a research academic at Adelaide Uni, so she's flown over specifically for this and to be with us today. Michelle has been very active in the eDGES project that I've been working on, and that's particularly now because it involves the Great Artesian Basin. So when this project was first proposed, it was looking at areas in the Pilbara, and then I was asked if I could involve the Great Artesian Basin, which I got very excited about, and thought I would reach out to the people who I knew had some previous work in that area. So Michelle has been working in groundwater dependent ecosystems in arid zones for the last 18 years. Michelle's been working very close with industry with her research, and has been involved in 4 previous ARC linkage grants. So that kind of demonstrates the value and the strength that Michelle has with working with industry and meeting research outcomes in industry questions. So Michelle and I started working together in 2017 on a previous linkage grant, and that was, can we even detect subterranean species from pulling groundwater? Do they even shed DNA into the environment, and can we detect that? That started in 2017. There's a new ARC linkage project underway, and we have the eDGES program going. So that was a big tick, a big yes. All right, so I'm going to pass over to Michelle, and she's going to give a little bit of a background to the Gap Springs and her involvement with those.
Thanks, Nicole. So I'm just going to give it a little bit of background on what Great Artesian Basin springs are. And for those who don't know, not everyone knows what they are. But essentially, they're springs that are groundwater fed from the Great Artesian Basin. The Great Artesian Basin covers about a third of Australia underground. So in the on the map, it's shown in the grey highlighting. And there are springs, and so essentially what happens is during the monsoon season in North Queensland, the water moves down the Great Dividing Range and gets pushed underground, and there are areas of upwelling throughout arid Australia. So basically at the base of the Great Dividing Range and then beyond. And so there are Great Artesian Basin springs or Great Artesian Basin fed springs in Queensland, New South Wales and South Australia. Queensland and New South Wales have highly impacted springs. They have exceptional biodiversity, but they're impacted by water extraction. There's a similar situation in South Australia, but the impact is nowhere near as great. And the springs are very well preserved currently. And so just for the South Australian springs, which are my area of expertise, we've got a number of major spring complexes, which are in the colours on the map. And the complexes represent many individual springs that are all clustered together. And then there are clusters of the clusters. So there are many thousands of springs, and they kind of fall out in these hydrogeological locations. The springs have probably been there for around up to 1 million years. But when I say they've been there, the geological crusts that we see, so what we see is when the water comes up, there is minerals and geology that comes up and forms this kind of mound crust on the outside of the springs. And then you get the classic kind of mound spring shape with the water inside. But the water itself coming from the Great Artesian Basin probably predates this well and truly. Historically in this area, it would have been warm and wet, and so there would have been wetlands and lakes all through this area. And it's only in the last 10 to 15 million years that we've seen these major periods of aridification, and we end up with these little oases in the desert as we see them now. And so my business in springs is during 2009, 2012, my colleague Nick Murphy and I worked on the Great Artesian Basin springs and the biodiversity that's found in the springs. Historically, we knew that there were animals that live in the water, and we knew that they were endemic, but we didn't know the extent of the endemism, and we didn't know the extent of the gene flow or the movement of the animals throughout the region when we imagine that we basically have these individual wetlands in a very dry environment. And so over that 2009 to about 2012 period, Nick and I sampled as many spring locations as we possibly could, and we observed we sampled them and then we genetically characterised them, and we observed exceptional levels of biodiversity, even between spring groups and spring complexes. And sometimes we even found individual species that were found nowhere else in the region or nowhere else in the world. So extreme short-range endemism. We also observed that there are ancient connections amongst the springs, and even with the animals, with those animals and the animals that we find in groundwater in the Pilbara and in the Yilgarn. the animals that live in these springs are nowhere near as charismatic and beautiful as the Pilbara Olive Python. So I'm only going to just show you one filter feeding thing. But they are incredibly special, and they tell a deep story about Australia's climatic history, but also the value of these springs as biodiversity assets. And we see the patterns of evolution over and over in all of the different taxonomic groups that we've examined. And these animals are also really critical to keeping the water clean. They're like pool cleaners. They keep the springs unclogged and flowing. So they're incredibly important. And so Morton asked the so what question. And I guess as scientists, but also as custodians and of the locations that we work in, but also that we impact, we recognise that there is a need to protect and understand the environments that we have in our backyard, and so we have Commonwealth legislation that protects the springs. The springs are specifically named under the EPBC Act as a significant biological community. And we also recognise that these springs have central value to the Indigenous custodians of this area. And so those two things make them incredibly important to protect. And we also need to understand and monitor them for the impacts that might be occurring. And so when we're, working on monitoring and understanding what species are where and what genetic diversity is where, it's incredibly time consuming, as you know. So we have identifying individuals, trying to figure out what they are. Most people don't know what they are because they all look like that little tiny thing, or smaller and tiny and green. And so there are very few experts now who can help us, and in terms of identifying it, and genetics can tell us a lot, but it's still incredibly slow. That whole process is really slow. And so as we will talk about today, Nicole and I have been working really hard in subterranean systems, but also now with the Great Artesian Basin Springs to develop up new technologies for monitoring and understanding the biodiversity of these incredibly important groundwater dependent ecosystems. And so in terms of the work that I'm doing, I'm still working on my subterranean work in the Pilbara and developing that up, but we're also moving back into the springs, which is really cool, with eDGES.
Okay, so I put this slide together. And for the non-molecular biology people in the room, we've kind of been throwing around some terms, and barcode has been one of them. So I thought, just briefly, for those of you who know what a barcode is, just please be patient. So as you can see up there, that little purple thing is usually the standard barcode. It's the CL1 barcode. It's come out of the Barcode of Life Consortium out of Canada. It's identified as this region as being the molecular identification tag. So if you want to ID something, you use primers, you target that area, you sequence it, you get the sequence, and then you can compare it to a reference database, or if you know what the species is, then you generate the DNA barcode. So it's very similar to going shopping in the grocery store. You pick up a brand of ketchup and you scan it on the barcoder. It's going to tell you the price and the brand of ketchup that you just purchased. This is a DNA barcode. Yes? Well, I wasn't satisfied with that. particularly with subterranean fauna. So beyond the barcode means I'm throwing out this little bit of DNA and I'm now targeting the complete mitochondrial genome, right? So rather than 600 bases, we're getting a lot more. So this has really been the basis of the project. There were some incentives with this because subterranean animals, they're really old, ancient lineages of animal and not the standard barcode primers work across because they're very different. They've evolved in isolation and they accumulate mutations, right? And for primers to bind on DNA, not to really give you a molecular biology lesson, but the primers need to stick to the DNA, and if there's too many differences, because the species are different, the primers aren't going to stick, and therefore won't be able to amplify. So I've gone with this approach. And as you've heard, we've got new sequencing technology in the block, but no one has introduced you into what we've named our new instrument, and that is DECA. For Ann Deca, who previously used to be the head of environment for BHP. I am very, very, very excited to have this instrument in the lab because this really upscales our ability now for R&D and for genomics. So previously I was going off campus to use an instrument like this to do my work. Now we've got it on campus and right in the lab, and I'm really excited. Okay, so what we have been talking about is subterranean fauna, and to give you an idea of some of the challenges, this is how small they are. Yeah, so I'm working on some things that are less than a millimeter to generate a shotgun library so I can produce those whole mitochondrial genomes, any additional information. So this has actually taken a little while to optimize in the lab. This wasn't a very straightforward plug and play thing. I had to optimize this because the kits that are the commercial kits that Illumina produces, they kind of specify you need a certain amount. It has to be high quality. You've already seen the Pilbara Olive Python. So I put an application into the zoo to do a high quality genome. We needed to bleed them. I had to get permission. I had to go straight on dry ice, very particular for the quality of DNA. I don't have that luxury with this project, but I'm very happy to report that it's working. Some challenges, not all of it works, but most of it works. So, and we are able to extract some really good information now when we're going at the mitochondrial level, right? So we're moving beyond that barcode. So with the shotgun sequencing, we're kind of able to make the reference mitochondrial genomes for the target species, and therefore then we can use that to develop our eDNA markers to target specific groups. So that's kind of what I've been working very hard on in the lab over the last couple of years to get that up and running. This slide shows Western Australia subterranean fauna that's been worked on and put through this pipeline, this shotgun sequencing pipeline. These are the groups of animals that have come from the Great Artesian Basin, the Gab Springs. Now, the main thing I wanted to highlight on this slide is you would originally look at the Gab Springs and go, oh, gee, there's not much diversity here, you know? But really, it takes a lot of highly skilled taxonomists, which we don't have a lot of, to identify these animals to an actual genus and species. So when I receive the animals, I'm hoping that they've been identified to the lowest possible. That's not always available. So when I received the animals from the Gab Spring, these are the 5 categories that I got told that these animals were binned in, versus some of these other ones where we have some more experts in WA working across those. Here, we're always talking about custom databases, reference databases. These are extremely important for eDNA work. But as we can see here, only about to 25% of the species, subterranean species, are actually described and have a reference barcode on GeneBank. There's a lot of work that needs to be done in this area. Also, new species are being discovered every day. They're not described yet. So anywhere you look, you'll find something new. That's just the way it is. And we're starting to look more underground now. Yeah. We're also working on 2 subterranean vertebrates, which is known as the gudgeon, which is a blind fish, and then the eel, the blind cave eel, which actually isn't an eel, that's a fish as well, but known as the eel. So they're also underground as well. Very, very small little case study getting into looking at the data. The data was pulled off of the supercomputer for mitogenome construction last week. So this is fairly fresh. Just to give you an idea, so ostracods are those round oval little animals that I just showed you before. This is kind of the area that animals and individuals were sampled. Michelle introduced you to the spring complexes. So this is in the Lake Eyre supergroup, which is only a small portion of the springs. And so I started looking at, well, the ostracod, so that tiny little animal. And I've just taken out for the non-molecular biology people in the room, this is what we call a DNA alignment here. And these darker areas are the bases that differ between the individuals. So these are identifying the differences here. So immediately when I started looking at this, I thought, well, okay, there certainly seems to be at least 2 groups of ostracods that I received for processing in the lab. So I started looking a little bit at these groups, because one thing I'm interested in is what is that kind of species cutoff and threshold? Short range endemism, as Michelle talked about, is very important. But also when we are doing environmental DNA work and we're generating ZOTUs, what is the threshold for subterranean fauna that we need to use for identifying groups, and how much variation within that can we accept before we're going to say it's something else? So one group had 1.5% variation within the sequences, and the other group had 1% variation. And when I looked from this group to this group, we can see we have 14% variation at the DNA level. But what I was a little bit surprised about is they didn't have like any geographical structuring to associate that with. So we were finding the DNA sequences of these 2 groups were shared at the locations. So I have at the top of the slide, cryptic or closely related. Well, I would say both, right? So we have these are closely related, and there's some cryptic nature in the DNA that we wouldn't have seen before, because remember, I just got those animals, they were round and oval, and it said ostracod. And that was it. So this is giving us some insights now, which I'm getting excited about, because when you're finally, your method works and then you start to generate your data and you see kind of the fruits of your labor, because most of the time in the lab, we're just moving liquid around tubes. It can be very unsatisfying at times. So this is more satisfying. Okay, so as I said, this project turned a bit bigger, COVID. So travel got restricted. I was able to get some water over from the Gab Springs. I was able to rejig my budget a little bit because I didn't have to travel, so I could spend that money on samples. So we've actually looked at 195 spring water samples and did metabarcoding with 18S while I was then waiting for the animals to be processed. So then the animals came over. Originally, I proposed I was just going to do 24 mitochondrial genomes from the Gap Spring. I got 100 animals, so I just kind of went for it. Yeah, so all the additional work there. On this, as we can see, Michelle was describing where the water comes from in these Gap Springs. This is rock, so some of this water is contained, and it's up to 2 kilometers down under the ground. That's why it's very, very old. So you have different types of springs, and then you have bores. And if you look over here, we have some eDNA results looking at some deep level water. And then the spring water is that surface water, right? And we're clearly seeing different kind of biological profiles and DNA that's found in there too, which is also very interesting. So quite a cool system, the Gap Springs. Okay, so Michelle and I currently collaborate on a few projects and co-supervise students. Michelle is running a current ARC linkage, which is supporting MELAD's scholarship. And one of the questions we're always getting is in subterranean systems, how long does the DNA persist if we're identifying these? So this is something that MELAD's working on, and he's also working on developing the marker for the gudgeon. So that's currently underway there. Jake Thornhill, he completed his honors. He was working on some Stygofauna and Barrow Island, and he's really excited and would like to continue on with a PhD. And then we have other grants. I've mentioned the ARC linkage, and we also host Julia Perina's ABRS scholarship through Curtin. And Michelle is just going to briefly talk about her work with the Arabana.
So Nicole and my collaboration is deep and wide-ranging. And so my work that has come from eDGES 1 Subterranean is work that postdoc Perry Beasley Hall and I have undertaken with Kim Solly from BHP. And the major outputs or achievements that we've had from that project was a collaboration with Arabana, who are traditional owners of the area to the east and southeast of Lake Eyre, where and also in collaboration with the SA Museum and Rachel King, we're describing new species of amphipod, which are one of the invertebrate endemics from springs that are kind of distributed all through the springs. And historically, everyone thought they were all just the same species. Turns out there's a bunch of different species. I can't say specifically because this is still not published, but, oh no, I have it right there. I don't have the species names though, but what we are planning is working with Arabana. We're going to use Arabana language words for species names of the amphipods that we found at these locations. So that's really, really special, and will allow us to tell some cooperative stories about biodiversity and also Indigenous culture in the region. In addition, our work with Kim also has led to 2 publications about springs and their biological and cultural significance. So the first is a paper about ecological, evolutionary and cultural significance of the springs, specifically in South Australia. And then we've also undertaken a major lit review of the biodiversity within springs, and we've basically done an inventory of everything that's listed in the literature time. So those are the main outputs that we've had. And we are now putting together an ARC linkage project proposal working on springs, and Nicole will be involved in that.
Yeah.
Okay.
Great. All right, Shane's bugging me. He's saying wrap it up, wrap it up. Okay, here we are. So eDGES version 2 is something I look forward to. I have some aims. It's 500 mitogenomes that I want to do for subfauna. That's just my aim. I think soil and microbes and subterranean fauna is an under-researched area, so I would like to explore that a little bit. And getting across the gudgeon in Cape Range and potentially the Pilbara with the new eDNA marker that's being developed by MELAT. So we've been talking a lot about the springs, but also Ethel Gorge is the other site. Matt's here, Matt from Stantec. He's still handing me over animals. So let's stay tuned, and I hope to be talking about that when we meet next again in a year's time. Thank you, everyone.
Great. Thank you very much, Nicole and Michelle. We're a little bit on time, so I'm just going to move straight ahead and introduce Professor Simon Jarman, who's going to be talking about 2 projects today. So improving sensitivity and specificity of eDNA tools for invasives detection and applying it to benthic communities associated with infrastructure in the Pilbara. Oh, how did that happen? Okay. I'm sorry, Mika. Let's just go to things. Oh, where is it? How did, my apologies.
I can talk about that one. Thanks very much, Shane, and thanks everyone for coming. So as Shane mentioned, I'm summarising 2 projects. So the first of these projects is a project on basically improving the DNA-based methods that we have for detecting invasive marine species and improving them both at the molecular level in the way that we analyse these samples in the lab, but also in providing the information that we need to do that well at an ecological level and to actually understand the diversity of organisms that we're looking at in the wild. The other project is eventually going to be applying this and other methods in looking at the benthic communities that are associated with the port and the surrounding area, the area surrounding the industrial part of the port in Port Hedland in the Pilbara. As you can see from the list of people who are on there, I'm a late addition to the team of only one of these projects, only project 5. Most of this work has been done by Ewan, Cheryl Lee, Justin McDonald and Fred Wells. So I'm not an expert in all of it, but this program is being funded by BHP partly because of the companies, I assume, desire to connect the community to the sort of research that we do. And personally, I find working on marine things is always really good for this because Australians have a strong connection to the ocean. We all live as close to the sea as we can get pretty much most of the time, but the ocean's not a permanent habitat for humans. We're not marine creatures. Our eyes don't work underwater. We need face masks to see things. Even our sense of touch gets disrupted by being wet. We need other tools to do these kind of things. So a face mask works for seeing things, but eDNA is even better than that. We can see even more using eDNA techniques than you could by scuba diving or snorkeling. It's a great tool for doing this kind of thing. And the second picture I've got there, so there's a picture of Cottesloe with obviously Australians getting close to the ocean. The second picture is a picture of the fishing wharf at Port Hedland, where we were sampling just a few weeks ago. We had a small part of the project was sampling water at the high tides, both at night and during the day, to look at diurnal variation in what was in the water column. And so each time we went down there to filter water, that's one of our team filtering water with one of these drills that I'll talk about in a bit. People would come and talk to us about it, ask what we were doing, and we would explain to them that we're doing a baseline biodiversity survey of this port area. And it was a really nice opportunity to interact with the public and tell them about this kind of work. And people respond to it really well. They really like it, especially when you can say, because Port Hedland's a weird port, because it's actually, the waters are in quite good condition. It doesn't have very many invasive species. So you can say, look, we're looking at what is here so that we can detect things if they are introduced later on, because there's constant shipping going in and out of there all the time. So it's a really nice thing to be able to work on. So panning back even further to think about eDNA that's in the ocean. The ocean is absolutely full of information. So this is just a random picture of a wave that I grabbed off the web. And if you look at that and estimate, say the face height of the any surface here, I'd say it's probably about 6 meters and the depth of field would be about 20 to 30 meters. If you do the back of the envelope calculations on that, and we have numbers that are reasonably good for this kind of thing, filter out all of the macroorganisms that are in it and sequence all the DNA that's in that volume of water, it comes out to be about 800,000 terabytes worth of DNA that's in the water. So what that means is that you can't sequence all the DNA that's in the ocean. Obviously, you would never try and sequence all of it. You'd take point samples all over the place. But to extrapolate that even further, if you were to try and sequence all the DNA that's just in the Swan estuary, that would account for all of the world's data storage capability. So there's a lot of information there. And what this means is that we need to make choices anytime we analyze eDNA. We need to make decisions about what we're going to look at. So the first choice that you make most of the time is what sort of collection method you're going to use, how you're going to sample the eDNA that's in the environment. And I've borrowed and there are lots of ways of doing this. Any of you who have tried to set up an eDNA project, and I'm sure most of you have because you're here at this meeting, have had to think about what sort of filtering things they're going to do, how you're going to sample surfaces, all those kind of things. And all of those things work to some extent. They all work in different situations in various different ways. And I've just borrowed a slide that my colleague Hayley Miller's produced from a paper we've just had accepted. And this is a nice slide. It's all electron micrographs of different sizes of filters. So 0.45 micron filters are the classic oceanographic filter that you use to differentiate between what's dissolved and what's not. And microbiologists have used it forever, but there are slightly smaller ones, 0.22 micron ones, and all the way up to 80 micron ones. And you can see in these electron micrographs, these are the things that you pick up. And none of it's DNA, of course, because that's even smaller than an electron micrograph can go to. We're not sampling naked DNA, we're sampling blobs of biosludge, which doesn't sound as good as eDNA, and eDNA is bound up within that. And these are pictures of that biosludge, basically at different scales. So you can choose lots of different methods for sampling eDNA or biosludge to get your eDNA. But really, the method should be appropriate to your question. And part of what I'll talk about here is the sampling methods, because they are important. And we've actually put a fair bit of work into developing rapid sampling methods that are fairly cheap and widely applicable, because another problem with eDNA analyses in the past has been that the rate of biological replication has been low, partly because of field sampling methods, but also because lab methods are still somewhat expensive. So there's a picture at the top right there of Shirley sampling one of the rock pools on a platform near Port Hedland, using a peristaltic pump mounted on the head of just a drill that you can buy from Bunnings, so that you could rapidly sample water. That's what we were doing at the fishing jetty in the previous slide. And you can take multiple samples in a very small amount of time, which gives you statistical confidence in your findings and makes all of this work far more relevant. The next step after the field sampling is that you need to make molecular decisions, so which bits of DNA to sequence and so on. We've heard about that in some of the previous presentations. But just to recap briefly on how DNA metabarcoding works, which is the standard DNA analysis method that most people use for broad-scale biomonitoring. Say you are trying to identify a fish species. This is an alignment of DNA sequences of a bunch of fish just in one family in Notothenidae. All of the top sequences on this Patagonian toothfish. And you can see that there's a sort of consistent area in that bit of DNA in the middle of it, bounded by a yellow box, that only occurs in that species, Patagonian toothfish. In other relatives, they have their own definitive sequences. The bits on the end are the bits that we can amplify via PCR or various other methods that are consistent and allow us to get to all of the bits in between. So deciding which markers to use is really how we get through that mountain of DNA that's present in all environments to get to the specific little bits that we need. And that's hopefully relevant to the next slides, because one of the things in Project 4 is that Shirley has been working on developing better methods for detecting invasive marine species. So the invasive marine species that have potential to be problematic in Western Australia have been assessed. This is an assessment of the phyla that they come from that I think Justin McDonald probably put together. And arthropods and mollusks came out as being the most likely phyla, or the most the phyla that had the most invasives within them that were likely to be problematic in Western Australian waters. But that's problematic because we know not very much about arthropods and mollusks in Western Australia. There are so many of them. It's a fairly undersurveyed place. It covers an enormous latitudinal gradient. So we go all the way from the Southern Ocean up to the tropics within Western Australia. And we have coastal and offshore waters that are very diverse and have a huge number of environments. So it's full of mollusks and arthropods that are native here. And we need to know what's native so that we can work out what's invasive, because many of these invasive organisms come from nearby areas that have relatives within Western Australia. So we need more information about that, which I'll talk about in a minute. But from the molecular point of view, what Shirley did was she designed some new PCR primers that got around some of these problems with just detecting the DNA from some of these species. So this is just one example. If you're trying to detect some of these invasive crab species, a very broad scale PCR primer set that can amplify from most things, but doesn't give very good species identification. This Pochon 18S one. So the sort of the standard way of describing these PCR primer sets, these tests for eDNA, is that the person who developed it, their name goes on it. So Xavier Pochon presumably designed that, and the 18S is just the bit of DNA that they're amplifying from. It can identify a lot of crabs, but not to species level. So it will detect crabs to family or genus level. But if you're looking for invasive species, that's actually not very useful, because you may be detecting native species that are in the same family as an invasive one. The next one over, the Luray CO1 primer set, will detect things to species level, but it doesn't detect everything. It misses a whole lot of things, and parts of the database don't have the species within it. And so this was the problem that Shirley was trying to solve with new primer sets that would identify species likely to be invasive in Western Australia to species level from these samples. And she certainly did. So she targeted a different area. And at the moment, it's just called new assays, but eventually, presumably, that'll have Shirley's name on it, I hope. So basically, I'm just going to summarize this slide verbally. But what she did was she developed two primer sets, one that worked primarily on arthropods, one that worked primarily on mollusks, mixed them together so that they could work in the same reaction. And what it did was it allowed us to detect far more of those groups than the generic primer sets that worked on a wider range of invertebrates, but weren't so well focused on those two groups that we were particularly interested in. So it was certainly successful. That's essentially what the slide says. It's just easier to hear it verbally. But then going back to the problem of species that are here in Western Australia, it's compounded by the fact that actually most species in the ocean have some sort of cryptic relative. So the rate of cryptic speciation in marine species is absolutely enormous. And it depends on how closely you look at the morphology, of course. But here's a good example with these bivalves, that if you look at the pictures of them up on the far right there, they look pretty similar apart from the colors being different. They're very hard to tell apart. And so what she and Fred Wells did was that they teamed up with colleagues at the University of Singapore to get reference material from Southeast Asia. Fred and Shirley collected reference material from the Pilbara and from the south coast of Western Australia, and identified a couple of species from this group in the Pilbara and one in the south coast that were endemic to Western Australia. And the reason that they were focusing on this particular group of bivalves is that one of the species within it is known to be an invasive, occurs in adjacent waters to Australia, so it was likely to come to Australia at some point. And if it did so, we wanted to be able to detect it. So we don't only need the molecular methods to be able to detect these things, we need the background information to be able to make sure that we're identifying the right thing when it gets here, which is what they were doing in this project, successfully once again. And there's a lot of other undescribed biodiversity within Western Australia, especially within mollusks. And I've got a few mollusk examples here that Shirley and Fred have worked on. So this was an example of an oyster species found in the Pilbara. Very small, very hard to find. So these are being sampled from the intertidal zone in mangrove areas. DNA sequences have been extracted from these. It's a new species, so there'll be a formal species description made from it, and the DNA sequences will be deposited in public databases so that anyone can use this information to identify these species further down the line. A similar situation with these intertidal oysters. So these are from, oysters are a classic invasive group of species. There are lots of different oyster species all around the world. Fred and Shirley once again went and did a lot of baseline biodiversity surveying to find out what species were present in the Pilbara, collected samples, described them, and have produced publications based on this. So unfortunately, the survey also didn't find Magellanic gygas, which is one of the invasive species that they were worried about detecting in this area. So all of this kind of baseline work really adds to our knowledge of what we have within Western Australia and allows us to detect anything that is invasive further down the line. Continuing that work, one of the invasive species, so Magellanic gygas, is farmed in Tasmania. So it's difficult to have a farm species that can also be potentially an invasive and farmed in the same country, essentially, especially when they're shipping between Tasmania and Western Australia. So there is clear potential for it to be an invasive species here. The Singapore, the Southeast Asian origin of this means that you need to go there to collect samples of it and describe the morphological and genetic diversity within that species from its native range, which may be different from the farmed versions as well, of course. So Fred and Shirley have gone and done that once again and worked with colleagues in Singapore to make sure they could collect across the range of the species and get a full description of it. And another mollusk species that they've looked at is this Mytella strigata, which is a Caribbean species and has been recorded in the Philippines. It's been also recorded in Southeast Asia and is a species of concern for invasion to Australia once again. The current work that they've done on this is restricted to Thailand, I think, at this stage, but there is potential for this to move to Australia. So we do need to know more about the diversity of this group within Southeast Asia before it potentially moves to Australia. So they've once again completed their work and have a manuscript that will be submitted to Bioinvasions. For future work within this project, the next thing that Shirley will be working on is trying to improve some of the traditional morphological biomonitoring methods that are used for invasive species detections in ports. So the standard methods, as I understand them, are to put down settlement arrays, and there's a picture of one here. So this is a complex set of surfaces on which organisms can, larvae of organisms can settle and grow. The standard way of analysing these would be that you'd put them in the water for a certain amount of time. Then once things have grown on them, you pull them out of the water and morphologically analyse what's in it. It's pretty time consuming. It's fairly expensive to do. These kind of surveys cost a lot of money. So the thing on the top right that has lots of algal growth on it is a passive eDNA sampling system. And what Shirley is hoping is that this may provide a replacement for the traditional settlement arrays. So that's being tested now. Fred and Shirley have also been working on barnacles and sea slugs. The recent trip we did to the Pilbara involved a lot of sea slug collection, and they're working with collaborators from the West Australian Museum and from taxonomic experts from the US as well on the sea slugs as well to identify these species. And I think the recent trip to the Pilbara actually brought back 3 species of sea slugs that have not been identified before. Is that right? Yeah, 3 genera. Okay. Yeah, 3 new genera. So this is just one example of the enormous amount of undescribed biodiversity that we have around the Western Australian coast. And it's great to have people starting to make a dent in that. So moving on to the survey of Port Hedland. So this is just a picture from Google Maps showing the area of Port Hedland. The main operational areas is around the township, basically, and just to the west of the township. The numbers on this map are the sampling sites that we went to during our trip, although I think we didn't go to 3, or maybe we did. Maybe the rest of the team did. I didn't go. So all of the other sites, we did go to all those sites. So the purpose of this survey was to compare community composition in the industrial port areas with the composition of the biological communities in the estuarine and inshore areas that are close to those areas. So essentially the relatively undisturbed parts of the ecosystem. We examined the depth structure of these communities. So we sampled surface waters, mid-water and bottom samples from all of the sampling sites that we went to. We also did some diurnal sampling at a couple of these sites to look at variation in what's in the water column at night and day, because particularly if you're looking for invasive marine species, many of them have larval stages that may only be night available. So if you're designing a future invasive species monitoring program, you want to know that you're going to be able to detect the larvae of potential invasive species. And the overall idea of this was that we're going to provide baseline biodiversity information about the industrial part of the port and the area around it so that we can detect future invasive species should they come into that area. And as I mentioned earlier, Port Hedland is a really interesting port because it doesn't currently have any known invasives in it. And given that this is one of the busiest ports in the world, it takes more tonnage of shipping than any other port in the world. That's really interesting. And maybe part of the reason for that is that the surrounding ecosystem is actually still pretty healthy. So once you go up into all the estuaries that are away from the main industrial port area, it's enormously biodiverse. The mangrove support a very complex and presumably very carbon dense ecosystem. And it's really fascinating to see. So part of the idea of this project as well is that we're characterizing that ecosystem because it may be providing some buffering against the invasion of external species that tend to thrive when there isn't a competition for them, when things are being cleared out by other processes. So it's certainly of value to investigate that thoroughly. But anyway, we sampled all of these sites. We used these peristaltic pumps mounted on drill heads to do a large part of the water sampling. And the point of these, so these are things that Shane and you and Harvey have been developing over the last half year or so, is that they allow you to sample water very quickly. So anyone who's done standard water filtering where you go and collect a litre of water and wait for it to filter through some sort of vertical pump, it's really time consuming, really annoying. If you have to take that stuff back to your hotel room or somewhere on land, some lab or something like that to do it, it involves carrying around large amounts of water, which is really frustrating. This allowed us to filter samples in the field very rapidly. Over the course of a week, our team of 5 people eventually took over 1,000 samples by this method and by another method that I'll show you in a minute, which is by any standard a very large eDNA survey. It's very rare that people get to this level of replication and sample as many environments as we did. So we're certainly excited to see the results of this. The other sampling method that we used, I have a video of this, which I think is, I'm not sure if it's embedded. I'll click it one more time. Oh, there we go. So we use these paint rollers, and this is Ewan using a paint roller on a stick, basically, in the industrial part of Port Hedland. And we use this for all of our surface sampling. And it's amazingly, even with this sort of 6 metre long pole, the little paint roller, that's a standard 100mm paint roller that you can buy from Bunnings. Works really well. You can rub that over any surface that you want to get to. You can sample to about 6 meters deep from a small boat, which means that you can do sediment surfaces as well as the surfaces of pylons, as we did. We did a lot of pylon scrubbing with this roller. The sides of ships, if you wanted to. We can do all of these things really effectively, really very quickly, and we collected a lot of samples this way. The other thing that we're hoping to do as this project extends is to start using an ROV deployable version of this. So if we can put these on remote operated vehicles, that will allow you to sample the underside of ships, which will mean that you don't need to put dive teams into the water to go and look at hull fouling organisms by eDNA methods. So the point of this survey is that we want to get an overview of the total biodiversity in the Port Hedland area as detected by eDNA. One of the other things we're hoping to get is an understanding of the role of artificial structures as habitat for whole communities of organisms. So in that, where we were filming you and scrubbing that pylon there with the paint roller, the fish community in there is incredibly diverse. It's maybe partly because people aren't allowed to fish there because it's an industrial port area, but those sort of artificial areas provide great habitats for fish and presumably many other organisms. We'd like to know what that is so that we can assign some sort of value to these artificial structures and what they provide as an ecosystem service, essentially. And of course, the overall aim of the project is to have an audit of all the species that are native to the area so that we can detect future invasions effectively. And combined with the sort of information that Fred and Shirley are getting on what is native and information that we'll have to expand upon because there are a lot of species to be identified, this will hopefully allow us to do it with more accuracy in the future. So thanks for listening to that. We're going for time, Shane.
Good. Yep. Time for a question or two.
Yep.
Great. Thank you. Did anyone have any questions for Simon? Thoroughly explained by the looks of things.
Fantastic. Okay, very good. All right, thanks.
Excellent. All right, we'll move on to Mika now. I'm sorry, not how those slides got mixed up. So we'll go, where are you? Up here. There you are.
Yeah.
There we go.
Thank you.
Fire away.
Well, fortunately, my talk should be quite short because I have not started yet.
Sorry.
So eDGES 6 is all about terrestrial biodiversity, and we're trying to look at terrestrial biodiversity across the tree of life. So in general, biodiversity is like one of the Sustainable Development Goals. It's valued by the UN, by different people. And biodiversity loss is one of the highest risk factors for human and global economies. It's also being valued more in terms of natural capital and natural assets. People are starting to think about how we can evaluate these things accurately. So a lot of biodiversity assessments, especially in the terrestrial ecosystems, tend to be labor intensive and have narrow targets. I'm talking maybe a plant survey and a bird survey, or a reptile survey with pit traps and plant survey with remote sensing, but they tend to have somewhat narrow focuses. eDNA is one of the tools that can assess quite well biodiversity across really broad range of organisms and groups, like bacteria, fungi, vertebrates, plants, all the stuff you can think of, basically, that we have references for. Which is why this is about across the tree of life. However, when you're looking at eDNA, there are differences between aquatic environmental DNA and terrestrial environmental DNA. So most of the talks previously have been about aquatic eDNA. And that's because pretend these little dots are DNA, or things that have DNA in them, as I think Simon was talking about, the little blobs. That's DNA, and water is a pretty good mixer of DNA. So you can take a sample of water and it'll have a pretty good snapshot of the biodiversity in the area. It has enough DNA and enough of the biodiversity that you can get a really good idea of the organisms there. And same with the sediment, it has enough of that there. If you look at terrestrial stuff, the analogy of water would be air, which does have DNA. You can get vertebrate DNA, you can get plant DNA, and you can get bacteria, but there's just a lot less of it. So while a liter of water can get you a lot of biodiversity information, for air you would have to have a pump going for a week, which is not the same. It's a bit more intensive, and it still doesn't have as much DNA as you would get from a liter of water. It also tends to be really patchy in terrestrial ecosystems. So you can have places where there's a lot of DNA, there's a lot of mammal DNA, for example, if you find a bit of scat that has been there for a week or a month, that'll have a lot of DNA of that animal that produced that scat, as well as what it was eating. Or tree hollows will have the DNA of the animal that was using that tree hollow as a habitat. Or burrows will have that DNA. But if you want to take just a random soil sample, the chances of having vertebrate DNA can be quite low because it can be affected by the solar radiation, by the soil microorganisms, and all the things like that. So when it comes to trying to do broad terrestrial biodiversity monitoring, the first step in this project is going to be all about the terrestrial eDNA sampling. So we have a couple of aims for this. Is first, to explore some of the spatial and temporal scales of terrestrial eDNA monitoring, talking different collection methods and different substrates, because we need something that's going to be able to distinguish between different land uses, be localized enough that we're not getting DNA from too far away that it's not relevant, and to be able to give us relevant biodiversity information. The other thing is that a lot of the sampling needs to be able to be done in a remote location, somewhere where you don't have necessarily fridges and pumps and electricity. And this is to make sure that it can be done in a standardized way in very different environments. So really the outcomes of stage 1 is all about sampling. We want sampling methods that are not dependent on cold storage, that are easily collectible by non-experts, that require minimal equipment, that are representative of local ecosystems, and most importantly, able to distinguish between land use types. So we're looking at broad biodiversity. This is not as targeted as what we were talking about with the Pilbara Olive Python. We know where the Pilbara Olive Python lives, rock pools. We take samples of water, take samples around the rock pools. Hopefully we detect the Pilbara Olive Python. But now we're trying to do broad biodiversity that represents an ecosystem. So we need to do some baseline research and development to make sure that we're getting accurate representations. The other thing we're going to be doing as part of the stage 1 is adding to those public reference databases of especially those key organisms that are especially indicative of ecosystem condition. So once we have our eDNA sampling set and we know exactly what we're sampling and how and all of that, and we're going to be using that sampling method to look at our case study ecosystem. So this is sort of stage 2 of the project, where we're auditing the terrestrial biodiversity across many different land use types of varying ecosystem condition. So the plan is for this to be done at the Olympic Dam area. It has a lot of different land uses, including arid recovery zone, which has fences such as these to keep out feral animals. It also has a lot of pastoralism, mining, the Gap Springs as well as more residential and urban areas. So we have a lot of land uses to choose from to start with our audit, make sure that all of our sampling methods and our eDNA and our assessment of the biodiversity can distinguish between these different land uses, so that in the end we can have a guideline, kind of a map, what to sample, how to analyze it, and what that biodiversity information means. So I'll show you in a second. Outcomes for this stage 2 is to have, ideally, proven protocols for standardized, third-party verifiable, high-throughput terrestrial monitoring. The gold star. Hopefully we get there. I'm sure we will get there. This is to help improve some of the data availability, help with our management and our conservation of these ecosystems. It gives us new metrics for target setting and incentivizes and evaluates the investment in restoration. And I've got this little graphic here. This is based on the taxon independent community indexes. This is one of our goals that we're hoping to reach. With all of that biodiversity information, we can get an algorithm that can create an index based on the biodiversity that we get, that evaluates in some way the ecosystem condition, or at very least the land use type. So as I have not started this project yet, so this is a snapshot timeline for the next 6 months or so. I'll be starting the contract part-time at the end of this month. I hopefully have the reconnaissance trips early next year with some pilot sampling and sequencing done by June, when we can hopefully start the next stage of the project. Any questions?
Thank you very much, Mika. Does anyone have a question for Mika before we bring in the last speaker? Eerily quiet. Okay. Wonderful. Well, as you would all know, we've got a session after this that we can, all the speakers will be coming along and we'll be, you can ask questions there. Sorry, I'll just skip ahead to Mattia now. Mattia, are you online?
Can you hear me?
We can. Excellent. Okay. All right, well, I'll introduce you. So we'll have Mattia Sacco, who's leading the third project, which is titled for this presentation, Unveiling the Functional Ecology of Hypersaline Lakes Through Multidisciplinary Approaches. He'll also have a guest appearance from a PhD student, Gonzalo Salazar, who's enrolled at the University of Antofagasta. Over to you, Mattia. You need to change the slides, actually, don't you?
Yes, I'll say click when you need to click. Is that okay?
Sorry?
To change the slide, I cannot change the slide from here.
There you go. I've clicked the slide.
Okay, great. So thank you all for having me and for bearing with me. I'm currently in Italy because I'm with a visiting researcher position at the University of Parma, with the broader goal to widen the network and gain expertise from advanced statistical modeling. So 2 apologies before I start. Sorry if my Italian accent skyrocketed suddenly. And also if you hear in the background a baby screaming is my son, so don't freak out. So this is the outline of the speech today. So I will start with a brief background on the importance of saline and hypersaline lakes. Then, even more briefly, I will outline the project design, and I want to focus on the latest findings of this project, which is an international project involving the University of Antofagasta, and specifically Professor Chris Harrod and his team. And there's a PhD student funded through this project, Gonzalo Salazar, who recorded around 8 minutes of this speech. I will conclude with an overview on some preliminary results on eDNA from the latest field trip in the Chilean Precordillera, and I will focus on the next steps and take-home message. Click. Okay, so the title of this slide is Ecosystems Worth Your Salt, mainly for 2 reasons broadly. One is the ecological importance of saline and hypersaline lakes. You can probably click now, Shane. As you can see on the graph on the left, the ecological functioning of these ecosystems is nothing but simple, and they host a range of biodiversity that is mostly unknown by the general public. Especially in terms of microbial diversity, there are actually models for life beyond Earth. And based on that microbial diversity, there's also rich macroinvertebrate community that is actually providing food for wading birds. In case of the Precordillera, Chilean Precordillera, for example, there's 3 endangered flamingo species depending on the food sources provided by these ecosystems. And also there's terrestrial biota depending on these ecosystems. Overall, the biogeochemical cycles linked to this biota are very interesting from an evolutionary perspective, given the adaptations that have been gone through during the last few millions of years. The other side of the coin is that these ecosystems provide very important ecosystem services. They're good for recreation. For example, the top left picture is about the Great Salt Lake in the US. There's a very important ecotourism industry linked to some, for example, salt lakes in South America. They are powerful in North Europe and in Russia for Artemia harvesting, which is a brine shrimp that is crucial for example, recreational aquariums. But more importantly, they're crucial for resource extractions, as we all know. So I put 4 points over there just to summarize the importance in terms of take-home messages over there. Nearly 1 out of 2 inland waters are saline. They sequestrate a lot of CO2. As I said, they sustain biota, and they are natural laboratory that are targeted by NASA, for example, for astrobiological studies. Click. As I said before, one of the most important ecosystem service provided by these environments is the resource extraction, and lithium definitely is the main character here. Australia is actually the world's greatest lithium supplier, and Chile is almost there. In Chile, there's a scrutiny at the moment in the Precordillera region, which we are targeting through this project. But if you click again, the framework becomes even more complex when we put in the picture finance dynamics. So at the moment, there's better no lithium miners because there's concern in the Chilean on the Chilean side. So overall, the picture is quite complex, and the ambition we have is to protect these ecosystems through the use of eDNA, as you will see through the results that we will show you today. But just these 2 slides were just aiming to give you a bit of a background on the complexity of this project as well. Click. This is very briefly just a bit of an ecological insight. The world is actually getting saltier, so the relevance of this project becomes even higher. If we consider on the right, for example, that the global acceleration of freshwater salinization is due to all the impacts that we produce to these ecosystems. So the amplification of this pattern is actually unequivocal. So understanding the dynamics, ecological dynamics sustaining biota in these kind of ecosystems, saline and hypersaline lakes, is actually probably targeting something that is very ancient, but is probably telling us a story about our near future. Click. This is, again, very briefly, the project design. We aim to use multidisciplinary approaches to unveil the ecological dynamics in hypersaline lakes specifically. As I said, it's a collaboration between Trent and the University of Antofagasta. The idea is to incorporate the 3 conventional disciplines of biology, hydrology, and geology with some genetics, eDNA, and DNA metabarcoding from gut contents, for example. We will also use trophic ecology and specifically stable isotope analysis to unravel the food web interactions of the biota inhabiting these ecosystems. And there's a bit of geochemistry in terms of the carbon flows and nutrients load in this very peculiar, from anthropogenic point of view, very peculiar habitats. Click. Now, over the next 3 slides, I will show some of the results of a satellite study that we were forced to start somehow due to COVID. This is an international project that started in 2020 in the middle of COVID. And as I said in previous years, this forced us to think out of the box. And we realized that Rottnest Island was a perfect natural laboratory for us to test some of the assumptions that we wanted also to investigate in the Chilean Precordillera. Rottnest Island, as we all know, is just 20 minutes away, and it has actually hypersaline systems that resemble quite nicely the conditions of the Chilean Precordillera in terms of water quality. So what we wanted to do, we wanted to have an approach that would target from microorganisms all the way to wading birds in order to understand the interactions of the biota inhabiting these systems. So if you click, one of the first outcomes of the project in 2021, we published this paper that I described in detail last year, where we targeted, together with some collaborators at Trent and outside of Trent, we targeted the microbial community in these 5 lakes that are the lakes that you cross, for example, if you want to go from one side of the island to the other side, for example, Salmon Bay. And through eDNA, we were able to unravel both the diversity and functions of the microbial community. And what was interesting was that these lakes, which are very close to each other, are actually providing different conditions and different communities according to the changes in alkalinity and salinity. This confirming that the microbial community is hugely adapted in this under these conditions and are quite diverse in an ecosystem that from purely an aesthetical point of view looks just like water and some brine shrimps. You can see that there's a lot going on, especially from a functional perspective, which is, to my view, the most interesting point of view. In terms of macroinvertebrates, if you click one of the other outcomes of this project, which actually got published this year, was about proposing an integrative approach to combine conventional techniques such as planktonic nets and the use of eDNA. So what we found out was that certain taxa were really good with eDNA and others were only detected with nets. So the take-home message of this paper was that we actually need to combine them as most of the time in terms of the eDNA field. And this is also fulfilling the second step of the story that we wanted to tell in terms of wanting to understand the biodiversity of these 5 lakes in terms of the invertebrate taxa. At the moment, if you click, we are transitioning towards the third ring of the chain, which is work in progress. We want to target the feces of the wading birds because they actually come all the way from Siberia most of the time to escape from the winter in the North Hemisphere. And there's some endangered species coming all the way to Rottnest. By targeting the feces of these birds through DNA metabarcoding approaches, we want to understand the role of macroinvertebrates and microbes as well in sustaining and actually driving these migration processes at the end of the day, because these 5 lakes and the other lakes on the island are actually providing the ideal environment for these species to thrive and survive at the end of the day. We will also want to test the use of an alternative substrate, which has been tested through other researchers at Trend for detecting species through DNA, which is spider webs, but this is still in progress in terms of theoretical design, and I'll keep you posted probably on the outcomes next year if this goes through. If you click. Now, this second slide, and after this, Gonzalo will take over on his outline in terms of the PhD and preliminary results. One of the other outcomes of the satellite project at Rottnest Island is more technical, let's say, more methodological, in collaboration with Matt Campbell and his beautiful expertise in bioinformatics and microbial ecology. Rottnest Island and the 5 lakes, actually more than the 5 lakes, but the Hypersaline lakes at Rottnest Island host very interesting microbial mats, which can be compared to some sort of microbial skyscrapers. As you can see in this graph, there's different layers with different metabolisms associated with different conditions. And this is an ideal test, an ideal laboratory, again, natural laboratory for us to test some platforms that have been developed during the last year in terms of functional genomics. I won't go much in detail because it gets a bit technical, but if you click one more time, we provided some details on the platforms that we wanted to test. So we basically used microbial math as a model to test the validity of the functional prediction of these 3 platforms, Picrus-T, Tax4Fun, and MICFunBread, which are metabarcoding based with shotgun sequencing, which we all know is more expensive, but also way more accurate. We use microbial math because they're relatively stable, and that's one of the reasons. And the other reason is that specifically for these 5 Hypersaline lakes, there's previous studies done by researchers at UWA. So we can compare the outcomes of the metabarcoding versus shotgun functional prediction over time, targeting exactly the same microbial math, which is quite unique. As you can see in this graph, very briefly, there's a set of metabolisms that were detected with both approaches, shotgun and the 3 platforms that you can see there. sulfur, nitrogen, methane, carbohydrate. Now, as you can see, the outcome is comparable from purely an aesthetical point of view on this bar plot. But one of the benefits of this test is also that some of these three platforms allow to use shotgun sequencing data generated previously to enrich the database that is used to generate this prediction. So without going too technical, the relevance of this is pretty much outlined by the sentence that we'll show in a second. If you click in terms of the one of the
Lost him. Is Darren still in? Oh dear. Shirley? Mika, could you just pop your head out and see if Darren's outside? Actually, I think if I...
Good evening, everyone. My name is Gonzalo Salazar.
We might just move on to
PhD
Gonzalo's
program at
presentation.
Postdoc. To put my presentation in context, I want to talk a little bit about the country where I come from. So Chile is a very important country for the lithium industry due to its large reserves, which are found in the Salares or Hypersaline lakes, of which there are approximately 50, and most of them are understudied. Most of the studies refer to strains of microorganisms extracted from these systems for biotechnological purposes, and there is little information about the ecology of these unique and extreme ecosystems. One group especially that has been poorly studied are the invertebrates of these systems. So, why invertebrates? Why are they are so important? Well, they are important in filtering sediments and the degradation of organic matter, and also the biofilm grazing. And also, they are very, very important as a source of food for many wading birds like flamingos and other migrational birds. And an important thing is their species richness decreases with increase of salinity. So they are very, very sensitive to these increases of salinity in some salares. Um, and we know very little about their diversity and ecological function in the Chilean hypersaline lakes. The aims of the project, so determine the community composition of invertebrates in hypersaline lakes. So we are doing that. Calculate the relative contribution of different energy sources to consumers, characterize the trophic ecology of invertebrates and flamingos and the trophic interactions, and develop an empirical approximation for studying food webs in this ecosystem. These three last aims are the next step for me in the next months. Since it is not possible to study all 50 salares present in the north of Chile, we had to focus on 3 salares that are important from the point of view of flamingo conservation. We have Salar de Tara, Puxa, and Puylar. Very important because a lot of flamingo feed and breed on these sites. These sites are very difficult to sample because of its extreme conditions. They are high altitude, okay, and also the access is restricted by some governmental institution like CONAF. So to enter these sites, we need the permits also for sampling these special sites. Okay, in terms of the methodology, this is a multidisciplinary approach. So we're trying to do a lot of different things to complement themselves. as a whole, like as I mentioned before. Okay, so I will conduct feeding experiments, also civil isotope analysis of invertebrates, DNA metabarcoding on gut contents of the invertebrates and flamingo feces. Also, I will do classical gut content analysis to compare with DNA metabarcoding gut contents. And also, the other part for assessing the community composition, I'll be using the eDNA and the classical invertebrate identification with the common net sampling and inspection in the laboratory. After a sampling we did in 2021, at the end of September and the beginning of October, we had some observations that are also results. So we can say there are preliminary results. And what we can say is each salar had very different characteristics in terms of the type of sediment, depth, and salinity. The type of organic materials present in the samples, also the invertebrate presence, the invertebrate taxa that I will show you in the next slides. And the other important thing is the abundance and size of invertebrates. For example, in Pilbara, there were a lot of amphipods, little crustaceans, but in the other sites, like Tara, for example, I found also amphipods, but not in the concentrations of Pilbara. In Puxa, in the first sample in 2021, I did not find amphipods. But recently we did a sampling in 2023, so this year, I found amphipods, but near the spring water, near the salar. We found a marked salinity gradient on each salar, so that's very interesting. And there is important contribution of water from springs in these salares. The other important thing is we have no evidence of the presence of Artemia at the sites. So in these 3 sites, we didn't find Artemia. Well, here are some pictures of the most representative taxa that I found in the 3 Hypersaline lakes, as you can see here. Okay. Okay. Here I'm showing a table with some descriptive data of the number of species of invertebrates in the 3 salares. And as you can see here, the first preliminary results shows that Salar de Tara has a higher species number compared to the other ones. And some samples remain to be examined from 2021. And we have new samples from a sampling carried out in October 2023. The next step to be performed with the samples are stomach content analysis and stable isotope analysis.
Okay, so I think that's the end of Gonzalo. Do we have Mattia back?
Yeah. Do you have 5 more minutes?
Yep, go for it.
Okay. For the screaming? Okay, so this is a bit of an outline on the theoretical framework and the preliminary results from Gonzalo's PhD. And as you'll see over the next 3 slides, the eDNA results are quite interesting in terms of matching what he found out through conventional approaches. So this is the summary of the field trip in 2021. We collected 27 sediment samples and 49 water samples. Today, as you'll see if you click, I'll focus only on water samples. What we did in both substrates, we targeted from prokaryotes to birds, so bacteria 16S for prokaryotes, algae, macroinvertebrates, and birds. It looks straightforward on a PowerPoint, but collecting samples from these places, if you consider that Salade, Puxa, is 4,500 meters above the sea level with very high UV radiation, probably one of the highest in the world, and very low temperatures. What these guys did was quite incredible in terms of collecting these samples and allowing us to have results for the first time for some lakes ever generated through molecular approaches. So let's have a look at the bacteria 16S prokaryotes results, just to give you a brief outline. Again, if you click, it should appear a simple bar plot for the three hypersaline lakes, Puxa, Tara, and Pilar. As you can see, Tara is the most structured in terms of the different phylums that we found. We used two different filters, 0.2 and 0.4. We separated the results here for prokaryotes, while for algae, macroinvertebrates, and birds, we merged the outcomes, the findings for illustrative purposes. What's interesting here, that there's a clustering definitely, which is more marked for Puxa and Pilar, while for Tara, the distinction in between the two different filters that we employed is not that clear. The next slide will briefly show you the findings for algae, if you click. And in red on top of the table, you have the total number of taxa found. As you can see, and the results are already matching from prokaryotes, what Gonzalo found through morphological approaches. Tara was the most diverse. This telling us that from an ecological perspective, the other groups apart from macroinvertebrates are also richer. So the entire structure is more complex at the side of the Tara, and the least diverse was Puxa, again confirming what Gonzalo found. The table could be kilometric, but I decided to focus on two charismatic groups. Navicola salinicola, as you can tell from the name, is a diatom, and from the name is abundant in brackish waters and inland salt springs. So again, we are using molecular approaches for the first time under challenging conditions because the UV is very high. Therefore, the preservation of the DNA is definitely affected. But through the pipeline we have in place, we were able to detect hosting biota of these ecosystems, and also Dunaliella, which is quite widespread all around the world, but is the classic algae that is referred as the group inhabiting the hypersaline environments, because they have an advantage from an ecological perspective that allows them to outcompete other organisms. Last but not least, Tara also had the highest diversity of cyanobacteria, which is frequently reported in the literature for hypersaline lakes, but as you can see, the difference is quite high, confirming again that Tara was the most diverse here. Then next slide will display some results about macroinvertebrates, which is probably the most interesting because we have the comparative aspect with what Gonzalo did. As you can see, the groups are quite similar compared to the table that he displayed. Tara was again the most diverse for CO1. CO1 wasn't fantastic in terms of copepods, as you will see in the next table, but I just wanted to reinforce that we did find Hylella, which was mentioned by Gonzalo, Puxa and Tara, and Hylobia atacamanensis, which is, as you can tell again from the name, is endemic, was found at the Pilar Lagoon. If we click next, we will display the 18S, and as you will see, there's a bigger diversity in terms of copepods, Cyclopodia, Aparticoida, and Calanoida. I wanted to stress again that Tara was the most diverse. We found Mesocra rapiens, which is found again in inland salt springs. And for ostracods, Darwinola stevensoni, which is a species that again is quite abundant in saline lakes. Last for this slide, if you click, I just wanted to highlight the importance of copepods. and the ecological relevance of them, because they're the ring connecting microbes and upper levels in the trophic chain. So what we found through eDNA is actually confirming that from an ecological point of view, these systems are nothing but simple. And the molecular tools that we are employing here are allowing us to unravel that complexity probably for the first time for systems like Tara and Pusa, which have virtually zero data on macroinvertebrate diversity. Next slide, just to conclude this section on the preliminary results, is on birds. We did find flamingos, which is already a big take-home message of the project. There's one species, Phenicopterus silensis, of the genus Phenicopterus. The other two species are Phenicoparus. We could not find them, probably depending on the fact that we rely on public databases. Therefore, Sanger sequencing and species-specific assays will be definitely more powerful. But broader, we did find avifauna that was cited in the literature as endemic and actually specific to those lakes, such as, for example, the Patagonian crested duck, or, for example, Spatula puna, which is another duck, and also wading birds, which are really good on migratory processes, such as sandpiper or the other species, the Corythocephalus ridibundus, which is again cited very frequently in the literature for these three lakes. So overall here, the take-home message is that we managed to detect bird in DNA hypersaline water, which is an achievement, but way more relevant than that, we did find species that were cited in the literature as inhabiting these systems, probably for the first time. Next. Very briefly, next steps. So we want to keep going, of course, on this powerful eDNA data, and we are at the moment writing a paper, and we crafted already a title for that. We want to dig into the isotopic ecology and the food-web interactions and gut content of, for example, flamingos, and increase the value of that meta-community analysis and ecological modeling by adding expertise and knowledge from other research groups across the globe. And I wanted just to leave you with a positive take-home message based on what you saw today in this presentation. So if you click, what we can easily say is that eDNA in saline, hypersaline inland lakes can provide a powerful tool to unveil biodiversity at an unprecedented scale and efficacy. There is definitely potential for it to become a routinary tool in surveys for industry and regulators, as we are seeing in the literature from other research groups and also from projects that we are trying to start in this respect. And with this I conclude, and the last slide will acknowledge the wonderful multidisciplinary team allowing all these great results, the publications, the conferences, and there's also a new set of collaborations as of probably 1 year ago on experts in artemia, experts in hypersaline lakes, in algae, which is a very important topic, and of course my visiting position here in Parma. Thank you very much, and sorry if I took too long. Cheers.
Right, that brings us to an end. Sorry, Mattia, we're running a bit short on time, but if anyone's got any questions, then please fire them back to us, and we can, unless anyone had a burning question, sorry. We can get them back to Mattia and get those answers back to you. But of course, everybody is going to be hanging around for a little while, and then of course we'll move down to the print hall shortly after. If you have any other questions. So if I dare and try and give you the elevator pitch on each of these different projects. So to summarise, in the Python project, we saw the first genome of the Pilbara Olive Python, which is the baseline for a series of novel new approaches for finding eDNA in environments. And this can lead to how we can find Pilbara Olive Pythons better, but of course that can now translate to other organisms as well. In the Subterranean project, we've heard how Nicole and Michelle have been sequencing whole organelle genomes with an idea of refining those assays to improve on detection on a really tricky set of organisms. to help their conservation into the future. And Simon, Shirley and Ewan's projects in the marine space, we've seen how eDNA analysis is being refined over sample types and molecular binoculars to an application of invasive species to improve the protection of our waterways, plus a great body of work on near and present threats and an enormous, possibly one of the biggest one-week efforts of collecting samples across Port Hedland. From ECA, we've had a taster and introduction on the concepts and challenges of trying to analyse a terrestrial ecosystem, which is going to be a super interesting project as that one gets underway. And of course, just now we've heard from Mattia and Gonzalo on some really interesting microbial and macro invertebrate analyses towards understanding the base of a pyramid, particularly in the hypersaline environments, which is fairly understudied and gives us a new baseline to start understanding the higher echelons of the tree, of the food of the tree of life. So with that, I'm going to stop talking. It's time we wound it up, but thank you very much for coming along. This is our third event. We'll be having one more next year, and then we'll be assessing this next year should be when everything comes to a crescendo. So we'll look forward to inviting you all back again next year. And otherwise, please stay around and we'll look forward to talking more. Thank you.